Thank you very much for joining today on behalf of the Biochemical Society and Portland Press. I'm pleased to welcome you to today's webinar, which is part of the Biochemistry Focus webinar series, which has been very good so far. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text. We welcome suggestions for future topics and speakers to feature in our webinar series. So that's something we're always looking for. If you're interested in suggestions, please see the Biochemical Work Society website for more information. So my name's Gary Scala, and obviously I'm a biochemist and a member of the Biochemical Society. And in this webinar, I will be hosting Dr. Annie Godwin, who will be introducing CRispR-Cas9, particularly as applied to Xenopus tropicalis and Xenopus laevis model system. So Annie started off with an integrated master's at Manchester. So after completing that, which was a BSc and an M-level qualification, she got an RA position at the European Xenopus Resource Centre in Portsmouth. She worked there for a couple of years, and that's where I first met you, Annie. And instantly it was obvious that Annie was really into the genetic side of things, and then that led to a PhD entitled Precision Modelling of Rare Genetic Disease Variants, which you then carried on as a postdoc, an MRC funded postdoc, and where you are now continuing to be with the EXRC. So Annie will be talking about some of her work, and that'll be touching on the disease variants, but mainly focused generally around use of Xenopus modelling with CRispR-Cas9. And I'm sure there will be lots of questions because this is a really hot topic at the moment. But can you please send in your questions during the talk via text or type them into the question box, and we will answer them at the end of the talk. I'll be curating the questions, so if several questions of the same type come in, I'll just ask it once. So over to you, Annie, and I'm looking forward to hearing your talk.
Thank you very much, Gary, and thank you for inviting me to speak as part of this webinar series and share some of our experience using CRISPR gene editing in the Xenopus model. So hopefully you can all see my screen. I'll be talking today about both Xenopus laevis and Xenopus tropicalis, and towards the end of the talk, the majority of the work will focus on this little guy here, Xenopus tropicalis. So I want to begin by briefly discussing and introducing the European Xenopus Resource Centre and highlighting some of our transgenic lines. Before we begin to consider some of the experimental design and how to conduct CRISPR studies in Xenopus. And I'll start with looking at experimental design, targets within a single gene, guide RNA design, considerations around mosaicism and experimental controls, before thinking about tissue targets, what advantages there are between targeting in the oocyte, the egg, or taking an organ-specific approach. For example, targeting specifically the germ cells or perhaps the kidney primordia. I'll quickly run over how we synthesize guide RNAs at the EXRC before beginning to look at how the scope is changing for analyzing CRISPRs and the emerging genotype and phenotype pipelines. And then I'm going to finish the talk with going over the success of a spin-out from the EXRC, modelling rare genetic disease variants, and begin to consider some of the precision techniques which could be applied in Xenopus, so base editing and prime editing. So for those of you that aren't familiar, the European Xenopus Resource Centre is based in Portsmouth in the UK. It's been going for about 16 years. And it's home to around 30,000 animals. And we currently estimate that we help around 200 labs worldwide that study Xenopus. The EXRC supplies live animals. It distributes animal products and DNA resources. For those of you that aren't familiar, the frogs are fully aquatic, as you can see here in one of our room setups. And the view that we have is a very nice view of the Spinnaker Tower compared to sort of the view that the rest of Portsmouth have, which is of this building here. This is where the EXRC is actually situated. And this slide here just gives you a bit of an example of some of what we supply and some of what we do. I should point out, if you are interested in purchasing wild type frogs, unfortunately due to COVID there is quite a long waiting list, which we're hoping to be on top of by autumn. So if you do want wild type frogs, please contact us sooner rather than later to let us know. So transgenic Xenopus are largely made for research purposes, to visualize gene expression in vivo across multiple time points. Largely, we take advantage of recombineering and use the p-transgenesis multi-site gateway recombination system in combination with ISCI1 to enhance that incorporation into the genome. Unfortunately, this does mean the transgene integrates randomly into the genome. So more recently, we are inserting some of the transgenes using CRispR into specific locuses. The EXRC is currently holding 139 transgenic lines, including this WIMP reporter line here and this oxidation state readout line. Our priorities for the foreseeable future were directed by the Xenopus community at one of the recent European meetings in Ghent and include the curation of an inducible Cas9 line, signaling reporter lines, and cell type labeled lines in Xenopus tropicalis. Again, if there is a transgenic line that you would like made, or similarly, if you have a transgenic line you would like to deposit, please get in touch with us via email. We're also happy to hold transgenic lines until results are published prior to distributing or advertising the resource. So what's the recent history of gene editing in Xenopus? Well, the first attempts to achieve sort of efficient, high throughput, target-specific gene editing in Xenopus were performed using zinc finger nucleases and talons. Both of these worked fairly well in Xenopus, as exemplified here in this transgenic knockout from the Haaland lab using zinc finger nucleases. And again, you can see here the disruption of noggin using talons by Lei et al. But both of these techniques were rapidly overtaken by the introduction of CRispR. For those of you who don't work with CRispR on a daily basis, you have a guide RNA that's complementary to your target locus that directs the CRiSPR-Cas9 complex to the target site. There are two conformational changes which occur, and that results in a double-stranded break. And then cellular DNA repair mechanisms subsequently repair the break. In embryos, this tends to largely be by non-homologous end joining, and this results in a variety of insertion or deletion changes in the animal. Typically, we've found across a number of CRispR experiments that the propensity of these indels is locus dependent. And we have repair outcomes which range from one predominant indel in single animals to being able to detect over 10, 15 indels in a single CRISPR embryo. But we are finding predictive algorithms are increasingly better at predicting these experimental outcomes, and we'll touch on some of those in a bit later on. I'm hoping to this audience that I don't need to sell the Xenopus model, but Xenopus are a powerful genetic model. They're small, they're easily maintained in the lab, and they breed quickly enough to facilitate genetic studies. There are now hundreds of knockout lines that are now both F1 and F2, and the EXRC alone is currently holding at least 56 mutant lines, and that's increasing week on week. CRISPR-Cas is so effective in frogs that almost all of the knockouts are sufficiently penetrant to allow phenotypic analysis in the F0. And here we can see the penetrance of 4 different phenotypes in founder animals, ranging from epilepsy to albinism, congenital cataracts, and congenital microcephaly. As reflected here, increasingly the diploid species Xenopus tropicalis is being used in these CRISPR screens over the allotetraploid species Xenopus laevis, particularly in disease modelling. And just to add another advantage here, these embryos are easily manipulated, they're growing rapidly, they're semi-transparent, and that facilitates a wide range of phenotypic analysis techniques. So when considering a CRISPR experiment, we must first plan where in the gene to target. Many CRispR experiments target the first exon or the promoter. As shown here, it can be a very successful strategy. We can see an experiment that's come out of the Granger lab here, where they did 2 sites in this paper, disrupted the promoter region and the first exon separately, and observed very similar expected phenotypes affecting the eye and the head in these tadpoles. Similarly, in this paper by Martin et al., who used constructs that were first published by the Miller lab to disrupt SLIC45A2, you can see the loss of pigment. And this experiment was actually done in an undergraduate practical, again emphasizing the power of these genetic screens, with benefits even extending into the teaching setting. That being said, it's important to point out that there can be cryptic promoters, especially when targeting novel genes. And unfortunately, the data showing the effect of new transcription start sites in these CRISPR-Cas9 experiments isn't being published. We do know from discussions within the community that people have faced difficulties when trying to truncate novel genes, but the instance of this remains unknown. And the impact is perhaps shown here in this paper by Gallegos et al., who show, using a reporter gene, that the first intron of UBQ10 can be used at a number of different locations to replace the transcription start site, and they additionally performed this in the background with a 303 base pair deletion that encompassed the promoter and all known transcription start sites, and they showed that that had no effect on gene expression as long as the intron was included. So to avoid potentially having a cryptic promoter driving transcription downstream, people perhaps would now look at early exons as an alternative, and this approach is exemplified by the Miller lab, who targeted exon 1, exon 2, and exon 3 in their preliminary experiments to knock out LHX1 when trying to study the loss of function effects in kidney development. In this experiment, they actually found that the guide RNA targeting exon 3 was the most efficient, and this allowed the group to investigate the developmental defects which arose in the CRISPR offspring, alongside the observed decrease in the kidney developmental markers, which can be seen here in the in situ hybridization. Unfortunately, this can be difficult to implement for genes with lots of small exons. So you're relying on a frame shift causing a premature stop codon, and when the exon is very small, often you can just end up with splicing out that single exon, and that might not have the effect that you're hoping for. And the other thing is to consider the effect on all the transcripts. Here we can see that DMD gene, we can see that targeting perhaps an earlier exon, we would still have some of these isoforms being expressed. So where early exons are not easily targeted, accessible by CRISPR, or where researchers are looking to create a second loss of function model to support previous findings in an early exon knockout, for example, can consider targeting key functional domains. And this again is shown here by Bluminx and NERC, where they describe how disruption of RB1 and RBL1 can result in the development of retinoblastoma in tadpoles, froglets, and frogs. And this was demonstrated in part by using a guide RNA targeting RBL1, which targets the pocket domain of the RBL1 protein, so one of its core functional domains. When using this approach, we must consider whether function may be partially retained, and also check for nonsense-mediated decay of the target mRNA, because that's going to be important in the downstream conclusions which are drawn from findings in the CRISPR model. So once we know where we want to target in the gene itself, we have to think about what would make an efficient guide RNA, and it's generally accepted that integrating CRispR-Scan and the Indelphi MESC model enables identification of efficient, high frameshift frequency guide RNAs in Xenopus. CRispR-Scan has actually become such a popular resource within the Xenopus community that it's now integrated into the tracks of the genome browser. And I actually understand that CRispR-Scan began in a lab situated in close proximity to a large Xenopus lab that was doing both basic and translational CRISPR studies. So they were able to access and also integrate frog CRISPR data into the development phase of CRispR-Scan. And we find CRispR-Scan very useful to identify guide RNAs with high cutting efficiency in vivo. We typically look for guide RNAs with a score greater than 50. However, we have had very good success with guide RNAs which have scores below 30. And we also use it to minimize or predict any potential off-targets. And we then couple this with Indelphi. So Indelphi we first came across due to Vlaming's and NERC, who raised it and suggested that they recommended guide RNAs would have a frameshift frequency over 75%. When we first read this paper, we went back through it and we did assess our previously designed constructs and found similarly that the guide RNAs with frameshift frequencies higher than 75% had a greater phenotype or more severe phenotype that had penetrated. In addition to this frameshift frequency here, we also look at the precision score and predominant indels, because this can be very useful for downstream applications, especially genotyping. So as we discussed in the earlier slide, we have seen a range from 1 predominant indel in crispant embryos all the way through to 10. And we're actually finding this model to be quite accurate at predicting how many indels we're likely to see and what they're likely to be. As a final point, there are many design tools that are available. CRispR-Scan and Indelphi are not the only ones that are out there, but please be aware that not all of them have the most up-to-date version of the Xenopus genome. And if you're starting out, CRispR-Scan and Indelphi are good because there's quite a lot of support within the Xenopus community for designing using both of those. So the classic way of creating a loss of function knockout model, as we've discussed, is by disrupting a critical exon and causing a frameshift mutation to induce a premature stop codon. But there are other equally effective approaches to disrupting gene function from CRISPR, including targeting either the donor or the acceptor of an exon and splicing out the exon. When doing this in the context of disease, just remember that splice boundaries can vary quite greatly between species. Using multiple guide RNAs to delete regions of a gene, we generally don't use multiple guide RNAs within the same embryo unless targeting 2 alleles in Lavis or performing a deletion experiment. And we have found that deletions work better if you first assess the cutting efficiencies in vivo separately and balance the amount of guide RNA that you're injecting. And to emphasize the point, so far we've managed to delete regions in multiple genes all the way up to 3 kb. I'm hoping you can just about see this band up here in this control embryo. That's the size of the target amplicon, and in each of these individual crispant embryos here, you can see, although there is a range of deletions that have occurred, we have got predominance where we've lost 3 kb in this gene. And we've also removed specific isoforms by targeting deletion to promoters. And just to make the point, if you are planning to delete a huge region, just remember there are, for example, enhancers or transcription factor binding sites within intronic regions, and to take that into account when potentially phenotyping the animals. And the final possibility, which is achievable using CRISPR technology, is inserting a precise template into the gene. people have inserted fluorescent proteins, or they've made deletions or point mutations using DNA donor templates. Here we can see a partial rescue of some tyrosinase mutants here using a phosphorothioate modified long single-stranded DNA template by Takuya Nakamura. And they actually reported seeing up to 35% rescue in the injected embryos. However, in our hands, we've generally found this approach to be quite inefficient in the embryo. And as such, we've moved to doing some of these insertions in the Xenopus oocyte. There are many advantages of the oocyte. The first thing is oocytes are full of HDR enzymes, which have decreased by the time it's an egg. The oocyte can be cultured for many days before being fertilized and division beginning, and this allows time for the gene editing machinery to degrade. So typically we found that guide RNAs and Cas9 are undetectable after 3 days in an oocyte. So that facilitates the creation of these non-mosaic heterozygous offspring with the maternal genome edited, and we can then produce embryos by ICSI. That said, this technique is very resource intensive, and it isn't sort of happening in many labs around the world. So if we begin to think around penetrance targeting tissues of interest, we must first consider that actually in some cases mosaicism isn't a big disadvantage, it can also be an advantage. Mosaic models often survive key developmental time points permitting downstream analysis. And we can take advantage of the frog's well-defined fate map to specifically target blastomeres up to the 64 cell stage, and this allows researchers to target, for example, the kidney primordia, and that enables the majority of the tadpole to be unedited, or areas outside of the kidney to have very, very low levels of mosaicism. But if you do want a non-mosaic model, it's possible to do this through breeding, because it is often necessary to overcome mosaicism. Traditionally, this was done by leapfrogging, but more recently, again, looking at the fate map, we know vegetal injections at the 32 cell stage largely target germ cells, or more recently, there are many labs now that are just injecting very, very low amounts of guide RNA at the 1 cell stage and finding that these much, much greater mosaic models are surviving through and able to reproduce. And we also must consider how to control for these CRispR experiments. So by and large in the literature at the moment, most people are now controlling their CRispR experiments using 1, 2, or 3 of these options. So it's very possible to target 1 of 2 cells in the frog. You can see here the first division is that left-right division, meaning that there's that internal control, allowing you to compare between each side of the embryo. You can use genes with known results, for example, tyrosinase, where you'd expect pigment loss, but perhaps wouldn't expect an eye phenotype or a craniofacial phenotype. Or perhaps more powerful is the use of multiple guide RNAs to target 1 gene. And this again is strengthened by targeting a different gene region within that gene, as we discussed earlier, perhaps targeting an early exon in a functional domain and showing the same phenotype coming through. Historically, and perhaps slightly less widely used now in CRispR experiments, are these rescue experiments. You can see here, this is a Cas9 control. You can see the knockout phenotype here in the eye, and you can see the rescue of the phenotype here. And we also have tissue grafts where you perhaps take and transplant across certain aspects of a mutant embryo, and you can follow that through in a non-mutant background. But as I say, these are being used less and less now to controlling CRispR experiments. So once we've got our experimental design and our controls in place, at the EXRC, as we say, we design using CRiSPR scanning and Delphi. We order standard single-stranded oligonucleotides from Invitrogen, and we follow the TAC-based method first described by Nakayama et al. To anneal and extend those single-stranded oligonucleotides into a double-stranded DNA template. We then use the Invitrogen mega short script kit to transcribe the double-stranded DNA into the guide RNA, which we aliquot at about 500 nanograms per microliter and store at -80. Typically, we expect a concentration range following an overnight incubation with this kit of around 1,000 to 3,000 nanograms per microliter. And we test a range of amounts preliminarily in both species, and that's seen there. We use a standard amount of Cas9. We use NEB's Cas9, which has 2 nuclear localization signals. And for genotyping now, we would recommend considering amplifying around 600 base pairs because we are seeing, although at low penetrance, larger CRISPR deletions that are coming through in a number of our models. So moving on to considerations around analyzing these CRISPRs. The first thing we must do is genotype the animals. And we strongly recommend looking at both the genomic DNA and the cDNA, particularly where guide RNAs are located in close proximity to the edges of the exon, a splice acceptor or a splice donor. We can see here in this mutant model that we have lost an exon, and we can see that again when we subclone and Sanger sequence this PCR product. We found in our lab that the T7 endonuclease 1 assay is a quick, rapid way to assess and indicate indels and samples. Again, here we can see a control PCR and a control T7. We tend to analyze these in batches of 5 or 10 embryos, and we can see here this assay recognizes mismatches in the DNA, and it will cut the band at the point where there's a mismatch, and it separates out nicely on an agarose gel. In cases where there is a target-specific antibody, a Western blot is a great way to assess protein expression in CRISPRs, although we are aware that we do need more good antibodies in the Xenopus field to target these locations. There's lots of discussion in CRISPR experiments around off-target analysis. Of course, when you're designing guide RNAs, if there is an obvious off-target hit, we would absolutely look at that region. But we largely rely on the fact that multiple guide RNAs producing similar phenotypes is likely to demonstrate an on-target effect rather than an off-target effect. I think probably it's fair to say most research labs are not in the position to store and analyze whole genome sequences. And actually, the models that have been whole genome sequenced and examined so far haven't actually shown that many off-target effects to be a particularly common problem in Xenopus. So what is possible with phenotyping CRISPRs? Well, there are, of course, the historic advantages of phenotyping in the Xenopus model. I think one of the greatest advantages in the Xenopus community is the support and the resources available through the various specialist centres located around the world. We have the ORFs through which we can produce thousands of probes for in-situ hybridization or constructs for rescue experiments. We have the well-defined fate map for targeting specific structures. And nicely well-defined experimental stages previously by Newcoop and Faber and now more recently updated in the Zahn drawings, just to name a few. And we can combine this with some of the well-defined and established experimental methods around DNA replication repair, cell cycle control, protein trafficking, explants, xenografts and transplants. And again, here you can see from our research lab a neural tube explant, which we've grown on a cover slip, and we're able to image neuron outgrowth, and we've done this in one of our neurobeta tubulin GFP lines, which marks differentiating neurons. But there are other very exciting phenotyping techniques which are actually coming through fairly recently. So analysing crispants has always been strong using imaging techniques. The transparency of developing Xenopus tadpoles means crispant tadpoles are amenable to many techniques. More traditional perhaps are looking at gross morphology under bright field. Here we can see a model of charge syndrome in Xenopus tropicalis. We can see microcephaly, we can see some craniofacial changes. We can also see that there's an asymmetry of the heart here. We can use our transgenic lines in these imaging, and we can also use the antibodies for immunohistochemistry and in-situ hybridization to look at time points during development. But more recently, there's been a greater depth and more people doing this greater depth of imaging. The two examples I've chosen here are micro-CT and light sheet, but you can see the real advantages here of looking at a micro-CT image and being able to see disruption of the different layers of the eye without the need to section and stain the eye. And these images were actually obtained using a contrast reagent with a heavy metal phosphotungstic acid. And here again, this paper was released recently by Soren Leinkamp and Thomas Schnedt, where they take and combine light sheet microscopy and deep learning to start to begin to provide a framework for higher throughput characterization of embryonic model organisms. And they've started to have several pre-trained networks and detailed instructions for applying deep learning from kidney development through to neural craniofacial. And this is very exciting because the prospect of AI in the future means the throughput of these techniques is going to improve greatly. And more recently, and quite close to my heart, is the ability to be able to do quantitative behavioral analysis in tadpoles. Typically, Xenopus haven't been in this quantitative behavioral field. It's been very much in mice and zebrafish. But recent work is now facilitating the screening of Xenopus models to identify phenotypic traits in models of neurodevelopmental disease. And the example I want to show you today is the free movement pattern Y maze, which actually probes short-term working memory and cognitive flexibility in all vertebrates. So this is actually an example here of a virtual reality version of this Y maze for humans. So this assay has been done in humans all the way through the vertebrate lineage and now in Xenopus. They have a much more simplistic Y maze. But here you can see two tadpoles. It is spread up in this Y maze setup. And similarly to all vertebrates, we see this predominance of right, left, and left, right. Now this assay is based on two-choice discrimination. And it relies on quantification of deviations from randomness in these search strategies. So you have a choice as you exit each arm, whether to go left or right, and these are pooled in groups of 4, and you can see then that produces this nice set of 16 tetragram. And black here is our control. And like I say, we've got a predominance of right, left, an alternating strategy, or left, right. And that is exactly what is seen in humans, mice, fish, all the way through. And we can see that when we use an NMDA receptor antagonist, MK801, that we've completely lost this predominance of this alternating search strategy. We've applied this in models of a neurodevelopmental disease. So this one was GRIA1, which is a subunit of the AMPA receptor, and these patients had a severe intellectual disability. And we've got 2 regions which we've targeted in these F0 tadpoles. We've got an exon 2 target, so an early gene, and then we've actually targeted a specific point where there was a homozygous patient variant. And we can see in both instances that there's a significant decrease between black, which is the control group, and either orange or blue, which are the two crispant groups. And we're beginning to do some work with a collaborator in Southampton to look at action potential firing and neuronal plasticity. And here again, we can see in a different crispant model, here's our wild type control. This is a 1 of 2 cell injected Xenopus tadpole. So we've got our contralateral and ipsilateral side, and we can see in the crispant side a decrease in the number of action potentials that fired in the neurons that were recorded from. So we're beginning to look at whether we can marry up neuronal plasticity, action potential firing to complement the behavioural findings that we're coming up against. And I'd also like to share with you some more recent data in proteomics. So in collaboration with groups based at Imperial and UCL, we've been looking into a gene called ACBD6, which is involved in myristoylation. You can see here that when we inject a high amount of guide RNA into these embryos, we see gastrulation phenotypes that occur. And what's exciting is that in collaboration with this group, we've been able to identify myristoylated proteins in tadpoles. Even more exciting is that this is comparable and similar to human findings. And we've seen a significant reduction in the enrichment of these proteins in crispant tadpoles. We can also see that ACBD6 loss negatively affects myristoylation. And you can see here that in both guide RNAs, 68 and 71, we can see that Marx is depleted. And that fits in with a publication in 2004, which shows an essential role for Marx myristoylation in gastrulation movement. And just to make the point as well that transcriptomic analysis in this F0 MyoD crispant revealed significant disruption in the expression of a target subset of genes. So this proteomic finding, similarly to the transcriptomic finding, suggests that although these targeted embryos are genetically mosaic for the target gene, there is significant disruption in the expression of the target genes or target proteins, supporting the power of genetic analysis in these founder models. And another thing which seems to be quite up and coming in the Xenopus field is doing small molecule drug screens in Xenopus. So Xenopus are actually very amenable to these high throughput drug screens and multi-well plates. And there are a number of projects now which are being funded not only to understand potential efficacy of drug compounds, but also to try and predict or understand the presentation of some of the unwanted side effects as well. There are some preclinical screening going on to look at, for example, cardiotoxicity affected to chemotherapy that's sort of marrying up with chemotherapeutics. Although there are a few Xenopus protocols exist for this, those that are developed in zebrafish can actually be quite readily adapted into Xenopus. And of course, it's worth mentioning that there are a number of pharmaceutical companies and the OECD that are doing these PHETACs, these frog embryo teratogenic assays, or the Xenopus embryonic thyroid assay, which is looking at metamorphosis to address these chemical toxic teratogenic effects of compounds. And I'd also like to sort of just finish the section by pointing out Cognizance, which is a sort of recently publicized platform for a method of rapid drug repurposing. And they started with a frog model of Rett syndrome. This was a Xenopus laevis model, and it's a F0 CRispR model. And what they've done is they've looked at predictive algorithms to predict small molecules which they think would be effective to disrupt MECP2 gene that underlies the condition. And they've compared that to the standard treatment that the patients currently receive. And they believe that they've managed to find a compound which appears to be more effective than the standard. It outperformed in terms of cognitive ability as well as lifespan, they've reported, and they're hoping to take that forward into trials. So I'm hoping, again, I don't need to sell the model organism to this audience, but how did we sell this famous model organism to clinicians? And I've put some of the points together of things that we really try to emphasize as we started to begin to work in the disease field. And I think it's particularly important to bear in mind that some of these rapid screens are really quite cost and time effective in the Xenopus model. So I want to sort of move on to the spin-off from the European Xenopus Resource Centre, which is aiming to extend the gene editing methodology that was developed in the European Xenopus Resource Centre. Within this this MRC funded grant, which is looking at the impact of mimicking human rare disease variants of unknown significance in Xenopus, and see if there's an impact in the ability to clinically diagnose patients. Just to give an example, we understand from our clinical collaborators that there are over 18,000 patients in the Wessex or Southampton Portsmouth area alone that are waiting for a diagnosis. As part of this project, these variants of interest are clinically led. So this is based on patients who present to clinics. We don't know what's going to be the next variant that's going to come through the door, what the next phenotypic presentation will be. So this is really driving the approach we're taking to be quite versatile. The novel genes and the homozygous truncation variants are the low-hanging fruit. And they're the low-hanging fruit because they're easy to model with the techniques that we've currently got. We can use CRispR to make these homozygous truncations. But more work actually needs to be done to address some of the missense variants and those that are presenting in a heterozygous or autosomal dominant fashion. So the power of collaboration is very clear. Early experiments that we did in the lab suggested frogs could be useful to clinical geneticists in interpreting whole genome and whole exome data. And when we were forming this collaboration, we were really keen to make sure that we were returning results quickly. And our first referral was we were asked to model a restricted cardiomyopathy in a gene called TNNI3, which is the inhibitory subunit of the troponin complex. And we were able to return results to the clinic in 2 weeks. So we targeted a CRISPR-Cas9 guide RNA to exon 7 of TNNI3. What we saw was stunted growth. We saw blood that was being pulled in the atria. We saw these abnormally restricted ventricular structures. We were able to see in micro-CT images enlarged atria with blood pooling in the atria again in our crispant tadpoles. So that was in 2018. To give you an idea of where we are now, we have looked at the phenotype disease alignment in 18 genes, and 17 associations align very nicely with the frog. We've made 6 precision patient models and 21 crispant models. We have 16 urgent genes currently on the waiting list with over 90 genes that have had to be deprioritized for modeling. And we now have an estimated wait time for modeling in Xenopus, which is over a year. This work is being really well received by peer-reviewed journals, and I want to encourage more people. More work needs to be done to address this gap in diagnosis of rare disease. But that said, the impact of CRISPR is of course cross-disciplinary, from classic developmental biology, the structure-function relationships of proteins, to rare disease, cancer studies. CRISPR has furthered understanding into so many biological processes. And there's great power behind replicating CRISPR findings in multiple model systems. And what we're finding increasingly through the work we're doing, that actually you do need to be replicating some of these findings in at least 2 model systems. But as I touched on a little bit earlier, the guidelines for determining pathogenicity of a variant in a gene that's set out by the American College of Medical Genetics, is well used across Europe too, demand unambiguous evidence of loss of function being causal, and/or a well-established functional study demonstrating the deleterious effects of variation at a known amino acid position. So you have to perfectly replicate most of these variants to be able to make an impact in diagnosis. And this high bar is critical for patient safety, but it has meant that fewer than 30% of sequenced patients have a clear molecular diagnosis. So where can we go next? And this is where I want to touch on some of the emerging precision modeling techniques, and I want to start with base editing. So base editors are these fusion assemblies that comprise of a Cas9 nickase, a cytosine or adenine deaminase, and a guide RNA. We're going to concentrate on BE4, the cytosine base editor that does a C to T change. Okay, so it changes the C to T, and the later base editors have got a uracil DNA glycosylase inhibitor, which has been added to prevent cellular repair of the uridine-guanine mismatch. And they also have reinstated catalytic activity into His 840, and that's to enable this complex to nick the non-edited strand. And that actually improves the efficiency a lot, because it encourages the cell to then use this as the repair template to get this TA modified DNA. And we've actually found that base editing is working very well in F0, Xenopus tropicalis. So here is one of our examples from the lab. Is targeting tyrosinase. It's a patient variant that we took from ClinVar to trial this technique. And you can see this C to T change here in one of the CRISPR embryos. Because tyrosinase we know is recessive, which means you need 2 altered alleles to see the phenotype. We have roughly guesstimated that about 50% of cells are bearing a homozygous change. So that's really great. But unfortunately, you are still dependent on an NGG PAM, and especially for this base editor, and that limits the number of missense variants that you're able to model. Although there are base editors that are now emerging which are sort of PAMless or can use a variety of different sequences to edit, but we haven't had a chance to look at how they work in vivo. And sort of in the last few minutes, I just want to introduce prime editing. So prime editing came out with the promise of being able to do any small DNA modification under sort of 30 base pairs efficiently without the need for HDR and cellular repair, etc. However, just to show you some of the data we've got so far in Xenopus, we've been looking within ABCC6 to model, just to insert really a stop codon. We've used the control to synonymous change. So here you can see the wild type lysine is AAA and the modified lysine would be then AAG. Although there's examples of prime editing working very efficiently in mouse embryos, we've trialed PE3, which is the system which includes a second guide RNA. So you've got your first guide RNA with your reverse transcriptase template, your primer binding sequence, which will come along and binds, and then the reverse transcriptase would come along and insert your change. And then you've got this second guide RNA, which nicks the opposite strand, again encouraging DNA repair according to the RT template, not the endogenous template. We've trialed both mRNA and protein, and as you can see here in these Sanger sequencing traces, when we use mRNA, we don't see anything, and unfortunately, when we've trialed using the protein, which has been produced by a colleague in-house, we're seeing a large amount of collateral damage, and by that I mean we're seeing big deletions in the target region around the site, and we haven't had much success in precisely replicating variants. And perhaps sort of the last point I want to make is actually, although we started with modelling using synonymous variants as controls, perhaps as a research community, we should be a bit more reserved about doing this and instead maybe look at known benign variants in human traces, or even variants SNPs with high allele frequencies. So with that, I'll quickly summarize. CRispR-Cas is a great tool for knockout. It's a tool for knock-in as well. And with that, we can look at basic biological research, disease models for research, as well as screen small molecules to treat these. CRiSPR experimental design is critical to the outcome and should be appropriately controlled. F0 studies in Xenopus are powerful, with novel emerging phenotyping methods supporting in-depth assessment of these CRiSPR models. Disease modelling to support diagnosis requires precision modelling. Base editing works well, as far as we can see in Xenopus, and wider applications may be possible using these emerging PAM-less base editors. There is power in replicating experiments in multiple model organisms. And with that, I'd just like to sort of acknowledge all of the people who have been involved in the work today. From this is our lab head and head of the EXRC, Professor Matt Gill, the disease modelling team, the EXRC team, our fantastic animal technologists who look after those 30,000 animals I mentioned earlier, and of course all of our collaborators which help further understanding into the phenotyping, genotyping, and ability to use CRiSPR in Xenopus.
Hello, well, thank you very much, Annie. That was really interesting, very powerful technology there. So if I can tell the audience that if you have any questions, please type them into the question box and I will read them out and ask Annie. So please, I'll give you a second there to type in any questions that you might have. And while we're waiting for any to come through, Annie, I've got a question.
Yep, sure.
So if you're modelling genetic disorders, obviously your primary interest is often monogenic ones, but looking towards the future, you might be interested in polygenic diseases. How hard is it using CRiSPR to hit multiple sites? Would you have to do everything sequentially, or could you do them in one hit with 3 guide RNAs?
So I think that if you wanted to do a quick preliminary study looking at the 3 different guide RNAs or 3 targets in one gene to see if you can get that phenotype association, that would be a good place to start. And then I do think it's worth investing the time, particularly if you're doing precision modelling, to model each of them separately and then cross them. But for anyone out there who is hoping to study polygenic disorders in Xenopus, there's some great protocols that are available through the Wilsey lab, that Helen Wilsey, who's actually studying polygenic disease in Xenopus with respect to autism, but she's already done a lot of that work.
Oh, that's fantastic. So something's coming through now. So you in Xenopus, you're injecting the guide RNA, is that correct, into the oocyte or the egg?
Yes, that is correct. Yeah.
And how does the Cas9? What is it, RNA that's translated, or do you inject protein? I mean, how you the RNA is obviously RNA. But how does the protein? How does the Cas9 get in there?
So traditionally, people were injecting Cas9 mRNA, but now it's fairly affordable and commercially available as a protein. NEB sells Cas9, and that's what we're currently injecting. It has 2 nuclear localization signals attached to it, and it works very efficiently in our hands.
Okay, just wait to see if any more questions come through. So the traditional method of knockouts in Xenopus, as I remember, was frying morpholinos. How has anyone ever done? Sorry, they must have done this experiment. Compare a CRispR-Cas9 knockout to a morpholino knockdown. How well do those overlay to each other using traditional methods for knockouts, or even RNAi, for instance?
Yeah, so I know through the EXRC we've worked with people who traditionally do knockdown experiments who have come to the EXRC to do their knockout experiments. And we have seen some which have aligned beautifully, and we have seen some projects where we've seen slightly different phenotypes, which is interesting, and I understand that that lab has gone to follow up on that. It's difficult to comment without knowing all of the genotyping to know how effective the knockout was in that scenario. But there definitely are labs out there comparing morpholino and crispants.
Just wait for anything else to come through. Done the right button. So the when you do your knockouts, how long does it take? There's a question here. How long does that take? We say very fast.
It is very fast. So for Xenopus tropicalis, typically so if you inject, they typically lay in the afternoon. About 1 to 3 a.m. they're gastrulating. By sort of 6, 7 a.m. they've gone through neurulation. And within 2 days after injecting, they've gone through organogenesis. They are swimming, responding to tactile stimulation. And for the behavior studies, we tend to do it at a little bit later stage and when they're free feeding tadpoles. So that's about 2 weeks, 2 to 3 weeks after they were initially injected.
Okay, we've got a question here. How do you see the future of CRispR technology as a therapy for treatment of disorders and diseases? So as opposed to a confirmation of the gene disease allele, but as actual treatment?
Sure. Yes, that's a good question. So there have been plenty of examples where they have corrected certain diseases using CRispR, or more recently, there's been some base editing examples. There was a little girl a couple of months ago who had modified T cells put back in to treat cancer, and she was otherwise determined to be terminally ill. So I think the future is big, and I think if we can get these precision techniques even better, that will just make a big difference to correcting disease in a more controlled fashion.
So I'll just ask the audience if there is any more questions. Well, I'd like to thank you again, Annie, for speaking today. That was a very interesting talk. You can follow the conversation online at BiochemSoc and PPP Publishing on Twitter, if you're a Twitter user. As I said earlier, we always welcome future topics to the Biochemistry Society for these webinars. And if you have any ideas, do please send them in. You can watch all this and all other previous webinars at www.biochemistry.org/events-and-training. So this and all others will be available to you if you're a Biochemical Society member. So please do have a look at that back catalogue. It's really good. And have a look for all upcoming seminars. So this one in particular, which I do recommend you watch again, will be available in a couple of weeks once it's been processed. Our next one will be on the 2nd of March at 3 o'clock, and it will be theoretical and practical aspects of CRispR genome editing in zebrafish. So a follow-on to frog CRispR-Cas9, and that will be Dr. Fredericus van Egden from the University of Sheffield giving that one. So that will be a great one to watch if you've enjoyed this. Finally, I would like to say it's been difficult over the COVID period. I mean, we've all experienced the problems of not having in-person conferences and meetings. Out of it, I guess this webinar series and things like it have been one of the most exciting things that have a good thing to have come out of it, and I do strongly recommend we all partake and make the most of that. And please, please, please do disseminate the information about the Biochemical Society and get friends, students and colleagues to join. It is your society. So thank you very much to everybody, and I think that's it for today. Thank you very much.